For decades, the medical establishment blamed autism on refrigerator mothers, a theory suggesting that inadequate, cold parenting caused the associated behaviors. Later, a completely baseless panic linked the condition to vaccinations. Autism is a neurodevelopmental biogenetic condition. It is a specific neurological architecture a person is born with, where neural pathways, like the ones tangling on the right, develop entirely differently than the standard paths on the left. 40 years ago, the incidence rate was estimated at 1 in 2,500. Today, data from the Centers for Disease Control and Prevention places the prevalence rate as high as 1 in 54. The condition is highly heritable. Data from twin studies reveals that if one identical twin is diagnosed on the autism spectrum, there is an 80% likelihood the other twin will receive a diagnosis as well. Despite this 80% heritability rate, genetic research has not discovered a single autism gene or a localized biomarker. Instead, scientists have identified over 100 different genes harboring mutations linked to the condition. To map how these diverse genetic variables create the specific behaviors associated with autism, we have to discard surface-level psychological profiling and examine the physical hardware of the brain from the synapse up. In the first 4 years, an autistic child's brain undergoes an unusual, rapid growth spurt. Scans show these brains are physically larger and heavier than non-autistic children of the same age. This animated diagram illustrates a process called synaptic pruning. Between the ages of 2 and 4, a typical brain culls irrelevant neural connections to increase efficiency. But in the autistic pathway shown on the right, this culling process is highly inefficient, leaving heavy, dense clusters of connections intact. Zooming into those dense clusters, we see the first half of the connectivity paradox, local overconnectivity. The brain produces an excess of short neuronal fibers, which act like short-range cables, amplifying signals that bounce rapidly between tightly packed nodes. Pulling back to a macro view of the network, the second half of the paradox appears. There is a distinct lack of longer fibers in the deeper white matter, the long-distance cables required to rapidly connect distant regions of the brain. Researchers at the University of Utah measured how these specific networks disengage from a task. They found that in an autistic brain, regions remain synchronized for up to 20 seconds longer than in a non-autistic brain. The research team compared this delayed shift mechanism to a telephone that simply won't hang up, severely delaying the brain's ability to shift its attention to a new stimulus. This architecture creates a severe neurological traffic jam. The brain becomes an operating system that is incredibly efficient at processing intense, localized details, but struggles mechanically to execute rapid, global shifts. These microscopic wiring differences scale up to affect the brain's largest anatomical structures, starting with the corpus callosum. The corpus callosum is the thick band of nerve fibers that acts as a physical bridge connecting the left and right hemispheres. This technical medial cross-section of the brain highlights the corpus callosum. MRI studies reveal that in autistic individuals, the axons within this bridge have a reduced diameter and lower volume, which directly restricts the velocity of signal transmission. Shifting our focus to the back of the brain, we find the cerebellum. Neuroimaging often shows this structure is smaller and less connected in autistic individuals. Cellular analysis shows the autistic cerebellum contains a reduced number of Purkinje cells. These specific cells are responsible for regulating fine motor coordination, balance, and building internal models of social behavior. Because the cerebellum's motor and social modeling circuits are under-equipped, an autistic person may appear physically clumsy or struggle to coordinate complex social responses. And with a restricted corpus callosum, the two hemispheres cannot efficiently synthesize broad, abstract ideas. The physical state of these structural hubs dictates the brain's baseline capacity. It places a hard biological limit on how fast distant information can be integrated to form a cohesive response. This top-down data model shows how a typical brain rapidly draws a bounding box around a chaotic scatterplot to identify a single recognizable shape. This represents strong central coherence. But as the model shifts to an autistic bottom-up processing style, it zooms aggressively into a single data cluster. It renders that one specific piece in flawless detail, while the larger shape completely fades away. This cognitive output is known as weak central coherence. In a practical setting, like a classroom, this piecemeal processing explains why a student might easily memorize vast amounts of raw data, but struggle to connect and organize those facts into a cohesive academic essay. This focus is driven by monotropism, a cognitive profile where attention is funneled into a single intense channel because the executive shift mechanism is rigid or sticky. When a monotropic mind is forced to process multiple simultaneous inputs, like listening to spoken words while tracking eye contact and decoding subtle body language, the system overloads. Behaviors frequently labeled as stubbornness or rigid obsession are actually the outputs of a highly specialized operating system. It prioritizes deep, single-channel systematizing over broad integration. One of the most complex integration tasks a brain performs is theory of mind. Theory of mind is a person's ability to intuitively recognize, track, and interpret the mental states, feelings, and intentions of others. Looking at this lateral view of the brain, the medial prefrontal cortex, located right here in the frontal lobe, serves as a primary node in the social brain circuit, responsible for perspective taking. Autistic individuals possess theory of mind, but underconnected social regions transform perspective taking into a manual cognitive task rather than a passive intuition. Predicting someone else's thoughts requires active mechanical calculation, a process of hacking out social cues. This active processing load frequently leads to a misunderstanding of empathy. Autistic individuals do not lack effective empathy. Effective empathy is the emotional capacity to genuinely care about another person's suffering, and autistic people often experience this intensely. The bottleneck exists purely in cognitive empathy, the mechanical ability to accurately identify and read those emotional cues or facial expressions in real time. While cognitive empathy involves reading fluid emotional states, systematizing is the exact opposite. Systematizing is the drive to analyze concrete rules, spot patterns, and predict exactly how a closed system will behave. Autistic individuals genuinely care deeply about the feelings of others. They are simply forced to manually compute and systematize complex social data that neurotypical brains are wired to process entirely in the background. Watch in this generated video how a single incoming pulse to this isolated auditory neuron explosively multiplies into a chaotic wave of signals. This happens because the locally overconnected autistic brain often fails to filter out unwanted background sensory information. Living inside this loud, unpredictable environment, the brain demands order. It seeks out strict routines, structures, and sameness to artificially generate stability it cannot organically produce. While clinicians categorize these behaviors as abnormal special interests, they function as essential biological coping mechanisms. Memorizing train timetables or categorizing fossils serve as soothing tools to extract coherence from sensory chaos. When these coping mechanisms fail in social settings, children adopt survival strategies. Psychologist Tony Atwood identifies 4 primary adaptations: internalizing self-blame, escaping into imagination, actively imitating peers, or outright denial. The imitation strategy is particularly taxing. Individuals carefully observe and mimic characters from television or popular peers, becoming highly accomplished actors to camouflage their autistic traits. This conscious performance causes massive cognitive exhaustion. The surface behaviors measured by standard diagnostic questionnaires, the routines, the mimicry, the social withdrawals, are desperate adaptations designed to mask an immense internal neurological load. This reliance on masking creates a severe clinical blind spot. Autistic females are consistently diagnosed years later than males, if they are diagnosed at all, occupying what clinicians call the invisible end of the spectrum. This conceptual matrix visualizes the gap between external behaviors and internal realities. Females typically rely on the imitation strategy, using stronger innate verbal abilities to mimic eye contact and deploy scripted responses, keeping their masking behaviors visible while hiding the internal costs. This intense impression management, often called camouflaging, allows an individual to superficially navigate social hierarchies, but the behavior never becomes instinctive. Because camouflaging is so effective, it successfully hides the underlying markers of monotropism and delayed theory of mind from standard observation-based diagnostic tests. When the exhaustion of performing this neurotypical mask eventually forces a breakdown, clinicians frequently miss the autism entirely. Instead, they misdiagnose the burnout as borderline personality disorder, severe anxiety, or eating disorders like anorexia. Our primary diagnostic tools are inherently biased toward the stereotypical male presentation of autism. To identify the condition accurately, clinicians must look past superficial performance and evaluate the invisible cognitive load. This historical photograph captures the intense, detailed workspaces of early tech pioneers. Environments like this drive a phenomenon known as associative mating, where individuals carrying genetic traits for high systematizing congregate and have children together. In populations heavily skewed toward STEM professions, like here in the Dutch city of Eindhoven, researchers have documented regional autism prevalence rates spiking to more than double the national average. This biological architecture routinely yields highly specialized skills. For example, Dr. Temple Grandin, a prolific author and livestock facility designer, has an asymmetrical brain with an elongated left ventricle and overconnected visual tracts, directly enabling her profound visual memory. Albert Einstein's brain autopsies revealed unusually large parietal lobes and an extreme excess of glial cells. These physical structures map directly to his profound visual, spatial, and mathematical capabilities. If science eventually identifies the exact complex combination of genetic mutations that actively trigger this specific neural wiring, we will face an uncomfortable ethical question. If we could intervene and normalize this architecture, would we actually want to? The autistic brain represents a highly specialized bottom-up neurological architecture. It trades global integration for profound local systematizing, expanding the collective capability of the human species.